Hello everyone, this is your ma'am Akshata and I'd like to welcome you all to the session today. And in our session today, we are going to talk about biomolecules. So without further ado, let's dive into our session. In our session today, we shall be talking about carbohydrates in detail, followed by proteins. All right, and then some amount of discussion will be there on vitamins and hormones. And finally, we shall be discussing about nucleic acids. Starting the discussion with carbohydrates. So what exactly is a carbohydrate? This is the definition. A polyhydroxy compound that has an aldehyde or a ketone functional group present either in free or as a hemiacetal or hemiketal is known as carbohydrate. Carbohydrates are substances having general formula CxH2O taken Y. Alright. So, these carbohydrates again are of three major categories, that is monosaccharides, oligosaccharides and polysaccharides. Talking about monosaccharides, a carbohydrate that cannot be hydrolyzed to simpler compounds is called a monosaccharide. Mono meaning single, saccharide meaning sugar unit. Monosaccharide which have 6 carbons are either aldohexoses or aldoketoses. Okay, they are either aldohexoses or ketohexoses. They can be aldohexoses or ketohexoses depending upon the functional group. If it is an aldehyde, aldohexoses. If it is a ketone, then ketohexoses. Example, glucose, fructose, galactose, etc. Talking about oligosaccharides, carbohydrates that yield 2 to 10 monosaccharide units on hydrolysis are known as oligosaccharides. They are further classified as disaccharides, trisaccharides, tetrasaccharides, etc. depending upon the number of monosaccharides that they hydrolyze into. Alright. The two monosaccharide units obtained by hydrolysis of a disaccharide may be same or different, and here are some examples of your disaccharides, which is sucrose, maltose and lactose. Now, what is a polysaccharide? A carbohydrate that can be hydrolyzed to many monosaccharide molecules is called a polysaccharide. A carbohydrate that can be hydrolyzed to many monosaccharide molecules is called as a polysaccharide. Example is starch and your cellulose. Now, what is aldoses? As I told you, monosaccharides containing aldehyde group are known as aldoses and the ones containing ketonic group are known as ketoses. So, aldoses have aldehyde group and ketoses have ketone as their functional group, as their carbonyl carbon. Alright. Now, both monosaccharides and oligosaccharides are crystalline solids, soluble in water and sweet in taste. They are collectively known as sugars. Polysaccharides are amorphous. They are sparingly soluble in water and they are tasteless. Polysaccharides are tasteless and they are known as non-sugars. They are tasteless. They don't have any taste per se. But these polysaccharides again are obtained from monosaccharides only. Monosaccharides polymerize and that is when you get your polysaccharides. Now, what is a reducing sugar or a non-reducing sugar? So, but sugars that are capable of reducing the Tollens reagent, the Felling's reagent are known as reducing sugars. Those which cannot reduce the Tollens reagent or the Felling's reagent are known as non-reducing sugars. Now, what you need to understand over here is all monosaccharides are reducing sugars because they can reduce both the Felling's reagent as well as the Tollens reagent. In fact, even fructose is a reducing sugar. Now, you will be wondering, ma'am, Tollens reagent and Felling's reagent is reduced by aldoses, by aldehydes. Then how come fructose is able to reduce? Fructose is able to reduce, beta, because in basic medium, it tends to exist in equilibrium with mannose with glucose, and all of them are aldoses and they can reduce it, right? That is the reason why. That is the reason why. All right. And why is it that they are able to see the presence of free carbonyl group? The presence of free carbonyl group is the reason why they are able to reduce it. Is the reason why they are able to reduce it. Now, one thing that you need to understand is all polysaccharides, all of them are non-reducing. All of them are non-reducing because their functional group is not free. It is not free for reducing the reagents. All right then. Now talking about glucose, CH2O taken, its formula is CH2O taken 6 times, that is nothing but C6H12O6. This is our glucose. This is our glucose. Okay, it is a reducing agent. Yes, it reduces both Fellings and Tollens reagent. What are its physical properties? It is sweet to taste. It is optically active and it shows mutarotation. The structure of glucose is like this. Now this is where it reacts with the OH group to give rise to the formation of hemiacetal. And it exists as pyranose. So this is in turn now OH is over here and OH is over here, so this is alpha. OH is this side to this OH, this is beta. I am writing the structure of alpha, H, OH, H, OH, OH, H, H, OH, CH2OH, H. This is alpha glucose. For beta around this carbon, the OH is shifted up and the hydrogen comes down. Alright beta, alright. So this is the structure of glucose, and this carbon, because around this carbon the orientation of OH varies. So this carbon, the first carbon is known as your anomeric carbon. Now what are the various chemical reactions that glucose undergoes? Now what a glucose undergoes reaction with acyl chloride. This gives us the information that there are alcohol OH groups that are present. It undergoes reduction in the presence of hydrogen, wherein the carbonyl carbon is reduced to alcohol. Then it undergoes reaction with CuO. Wherein the CH2OH, that is the free primary CH2OH, undergoes oxidation to gluconic acid. It undergoes strong oxidizing agent where both the aldehyde and the glucose over here converts into carboxylic acid. CH2OH converts into carboxylic acid. And finally, Osa-zone test is given. This is the Osa-zone test. So these are some reactions that glucose is capable to undergo. Now, what is epimers? Epimers are a pair of diastereomers that differ only in the configuration about a single carbon atom. Now, glucose and mannose are C2 epimers. This is the first carbon, this is the second carbon. This is glucose, right? Your mannose is going to be 2, 3, 4, 5, 6. Your mannose is going to be this. Now, you see, around the second carbon, this is the first carbon, this is the second carbon. Around the second carbon, the configuration is different. They are known as epimers. Rest all remains the same. Alright. Now, the cyclic structure of glucose. Now, why is it that we need to follow the cyclic structure of glucose, beta? The chain structure of glucose proposed by Bayer explained most of the properties, but it could not explain the fact that it does not give Skiff's test with Skiff's reagent, and it also does not react with NaHSO3. Now, these two reactions are what aldehyde group is supposed to undergo, but that it is unable to because it is undergoing acetal formation. Alright. Now, pentacetate of glucose does not react with NH2OH group, indicating absence of CHO group. hydroxylamine. That is why cyclic chain was explained. Now, mutarotation of glucose, the spontaneous change in specific rotations of an optically active compound is known as mutarotation. Optically active compound is known as mutarotation. So, does glucose show mutarotation? Yes, glucose does show mutarotation. Okay, it does show mutarotation. It does show mutarotation, beta. It does show mutarotation between the alpha and the beta forms. You see, between the alpha and the beta forms. This is the equilibrium mixture. Between the alpha and the beta form, it does show mutarotation. Plus 1193. At equilibrium, it is at plus 52.5. Right? So, this is mutarotation. Alpha glucose is plus 111. Beta is plus 19.3 and equilibrium is intermediate. These properties can be explained by monosaccharides. These properties can be explained by cyclic structure of glucose. Glucose forms a stable cyclic hemiacetal. Initially, 5-membered ring structure of glucose is proposed and it is known as furanose. Not furanose, pyranose. For glucose, it is pyranose. Okay, initially furanose, later pyranose was given. All right. Talking about anomers. Anomers are diastereomers that differ in the configuration at the acetal or hemiacetal carbon atom of a sugar in its cyclic form, or anomers are epimers whose configurations differ only about the first carbon. Okay, now fructose, it is also known as alpha levulose because in the natural form it is levorotatory, that is it rotates in the anti-clockwise direction. It is pentahydroxy ketone and shows mutarotation like glucose, and it shows mutarotation like glucose, and it is a reducing sugar. This is the structure of fructose. This is the structure of fructose. So if I have to draw the this is alpha. This is the alpha structure of fructose. Keynote. Unlike other ketones, fructose can reduce Fehling solution and Tollens reagent. It is probably due to the formation of an equilibrium between glucose mannose and fructose. Now, what are epimers? Diastereomers with more than one stereocenter that differ in the configuration about only one stereocenter. Only one stereocenter are known as epimers. Example, glyceraldehyde and L-glyceraldehyde. Okay, they are enantiomer as well as epimer. Erythrose, threose, glucose, galactose, they are all C4 epimers. Now, what is sucrose, beta? What is sucrose? Sucrose is nothing but when glucose interacts with fructose, what you get is sucrose. Glucose along with glucose in the presence of, or glucose when it reacts with fructose, what you get is sucrose. So, alpha glucose and beta fructose, when they come together, what you get is sucrose. Alpha glucose and beta fructose, when they come together, you get sucrose. So what does it look like? So it looks like this. Alpha glucose, HOH, HOH. This is your alpha glucose. Now talking about your beta fructose, This is going to be by the loss of water. by the loss of water, beta. Yeah. You will get sucrose. And rest all remains the same. This is your sucrose. This is the glycosidic bond. This is the glycosidic bond, beta. Glycosidic bond. And sucrose is a non-reducing sugar because the anomeric carbon are utilized in bond formation, as you can see over here. Maltose. Maltose, beta, maltose is formed when alpha glucose reacts with alpha glucose. That is two alpha glucose when they react, you will get mannose. You see the anomeric carbon over here is free. That is why it is reducing. Okay, it is C1C4 linkage. Okay, C1C4 linkage, and yes, it is a reducing sugar. Lactose is formed when lactose is formed when glucose and galactose form together. When beta glucose interacts with beta galactose, you get beta glycosidic linkage. You get beta glycosidic linkage. You get beta glycosidic linkage, and since the anomeric carbon is free, thereby it can undergo reduction. Talking about starch, starch has two groups, amylose and amylopectin. It has amylose and amylopectin. Amylose, beta, amylose is water soluble and amylose is not branched. Amylopectin on the other hand is water insoluble and it shows branching Amylose has a simple C1C4 glycosidic linkage, whereas amylopectin has C1C5 C1C6 glycosidic linkage. So you see C1C4 glycosidic linkage is there between the first carbon and the fourth carbon. Whereas when you look at amylopectin, it has branch between C1 and C6, and because of this heavy branching, it is insoluble in water, beta. It is insoluble in water. Cellulose, again, it is a non-reducing sugar and it has beta glycosidic linkage. Now, what about Tollens reagent and Benedict's reagent and Felling's reagent? Let me tell you, Tollens reagent, Felling's reagent, Benedict's reagent, all the three of these reagents are happily reduced by aldehydes. But they are not reduced by ketones. Fructose is an example, because fructose is capable to exist. It is capable to exist in the form of glucose or mannose in basic pH. It is in equilibrium with those forms. That is the reason why it is able to reduce these reagents. Now, what happens when Tollens reagent is reduced? You get silver mirror test. Benedict's reagent, when you get brick red. You get brick red color because the copper is reduced. Similarly, this is what happens. You get brick red production. Right? So all monosaccharides are reducing sugars. All disaccharides are reducing sugars except sucrose. This is one thing you need to know. Okay? And all polysaccharides are non-reducing sugars. You can get MCQs based on these. So the different kinds of glycosidic linkage is present over here. It is given over here. You can go through this for your understanding. Now talking about amino acids. See, beta, we say protein is the building block of the human body. But let me tell you, these proteins again are formed from various amino acids coming together various amino acids coming together by the formation of peptide linkage, by the formation of peptide linkage to give us peptides. These peptides again come together, and that is when we get the protein, and that is when we get the protein. So amino acids give rise to peptides, peptides give rise to proteins. Similarly, proteins on hydrolysis will give rise to peptides, and peptides upon hydrolysis will give rise to amino acid. And how is an amino acid? Amino acid is nothing but the carbon is there, an amino group is present, NH2, carboxylic group is present, hydrogen is present, or I'll write R, and any other group is present. This any other group is present. This can be any other group. This is your amino acid, amine group as well as an acidic group. This is your amino acid. Now, amino acids are again of two types, essential amino acids and non-essential amino acids. Amino acids which are necessarily to be present in our diet are known as essential amino acids, and they are not formed by our body. We have to take them in our diet. Non-essential amino acids they are formed in our body. They are formed in our body. Okay, they are formed in our body, and you don't have to take them through diet. But that is why they are non-essential amino acids, because they can be synthesized by the body. Now, some examples of essential amino acids are leucine, isoleucine, lysine. Some examples of non-essential amino acids is alanine, aspartic acid, aspargine, cysteine. Some basic amino acids. Now, amino acids are also basic and acidic. That means if the NH2 group is more in number, then it is basic in nature. If the COH group is more in number, then it is acidic in nature. So, lysine, arginine, okay, histamine, they are basic amino acids, whereas aspartic acid, glutamic acid, all of them are acidic in nature. Now, when we talk about optical activity, except glycine, all other amino acids are optically active. Glycine lacks chirality. Because the structure of glycine is this: NH2, CCOOH, H. Now, this carbon is not chiral. It has two same groups on it. That is the reason why glycine is the only amino acid which is optically inactive. When we talk about Zwitter ions, beta, this NH2 and this H, there's a very high probability that the exchange can take place to give rise to NH3 plus and CO minus. This is nothing but the Zwitter ion. Now, how is this peptide linkage formed? The part where NH2 is present is known as N-terminal. The part where the COOH is present is known as the C-terminal. And by the loss of water, you get peptide linkage. Alanine. By the loss of water, you get peptide linkage. In the above structure, COH group is C-terminal residue and NH2 group is N-terminal residue. Okay. Now, these proteins, they are high molecular mass complexes, and they are biopolymers of amino acids. Okay. Now, they are again of two types, fibrous and globular. If they are in the form of fibers, keratin, okay, collagen, all of these are fibrous, whereas globular is insulin, albumin, thioglobulin or hemoglobin, all of these are globular. That is, they are present in the form of globules. Structure of the protein is again of four types: primary, secondary, tertiary and quaternary. Now, when we talk about primary structure, it is simple structure, but a single polypeptide chain is present. That is it. But when we talk about secondary structure, now there is alpha helix that is seen. Okay. And then you have beta pleated sheet. Okay, the beta pleated sheet, it is like this. This is what it looks like, beta pleated sheet. Now, there are intermolecular hydrogen bonds that are present. Here also intermolecular hydrogen bonds are present, but between the two chains. In this case, beta, the intermolecular hydrogen bonds is present in the same chain. Here between the two chains, that is beta pleated sheet. Example of alpha helix is myosin, keratin, beta is silk and fibers. Now, what about tertiary structure, beta? What about tertiary structure? Tertiary structure refers to its three-dimensional structure wherein the beta pleated sheet and the alpha helix are present in the form together, like the complete like a ball together. That is nothing but the tertiary structure. Okay, it is basically like this. It is basically like this, like this. This is your tertiary structure. This is your tertiary structure. Okay, and this structure is formed by four types of bond: hydrogen bond, hydrophobic bond, ionic bond, and disulfide bond. All right, bete. Quaternary structures. Now, when two or more polypeptide chain units by forces other than covalent bonds are present, then we call it a quaternary structure. The best example is hemoglobin, where the alpha chain and the beta chain are present like this. So when a protein, now what happens is, what is denaturation of protein? Now what happens is, these proteins, beta, they have hydrogen bond, disulfide bond, but these bonds are very sensitive. All right, the moment you increase the pH, raise the temperature, these bonds break, they denature. That means the protein loses its tertiary and secondary structure. It is only present in its primary structure. Since the secondary and tertiary structure is destroyed, which is actually important for the activity of the protein, especially in the case of enzymes, okay, what happens is they denature. Primary structure remains intact, but the secondary and tertiary structure is destroyed. Curdling of milk, coagulation of egg white, all of these are examples of denaturation of proteins. Now, what are hormones? Water soluble amino acids are known as hormones. Okay, now these hormones are of different types. You can see epinephrine and adrenaline. They are used for controlling blood pressure. Okay, it helps to release fatty acids from the glucose. Okay, then and get glucose from liver. Now, thyroxine is secreted by the thyroid. It is very important. It is very important for metabolism. Then testosterone is there. Testosterone is found in the males. It stimulates the male sex organs. All of these are nothing but the various hormones that are there. All right, other than the hormones, we have vitamins, and these are defined as a group of biomolecules that are required in very small quantities. That is, vital amines are nothing but vitamins. Okay, that is nothing but vitamins. Vital amines are nothing but vitamins. They are again of three types. One is water soluble, one is fat soluble, and the other is neither soluble in water or fat. Okay, water soluble are B and C vitamins. Okay, B and C vitamins. Fat soluble is A, D, E, and K. And biotin, that is vitamin H, is neither soluble in fat nor in water. Now the problem is, beta, they are required in very small quantities. But if these quantities are not met, they cause diseases. Some example is vitamin A, also known as retinol. It causes xerophthalmia, cornea night blindness, xerosis. Okay, vitamin B1, beriberi, loss of appetite. Vitamin D, rickets, osteomalacia. Vitamin C is known as ascorbic acid and causes scurvy. All right. Now, the last group is nucleic acids. You know, DNA, RNA, they are both your nucleic acids, right? But how are they formed? When the nitrogen bases along with the sugar and phosphate bond come together, what we get is our DNA and our RNA. All right, they are DNA and our RNA, and they are also known as, these are obtained from nuclear proteins. All right, pentose nucleic acid or ribonucleic acid, deoxypentose nucleic acid or deoxyribonucleic acid is nothing but a DNA. Nucleic acids can be hydrolyzed in stages to get nucleotides. Okay, nucleosides and phosphoric acid. Nucleic acid gives rise to nucleotides upon hydrolysis. Nucleotides gives rise to nucleosides. Okay, base and sugar forms nucleoside, beta. When base and sugar reacts with phosphate, you get your nucleotides, which gives rise to nucleic acid. The two sugars are found to be present in nucleic acids are nothing but D-ribose, that is in RNA, and 2-deoxyribose in DNA. The bases, important bases are purine and pyrimidine. Purine are guanine, adenine and guanine, while pyrimidine are uracil, thymine and cytosine. Uracil is seen in RNA in the place of uracil. is seen in RNA, thymine is seen in DNA. Adenine, guanine and cytosine are present in RNA as well as in DNA, while thymine is present only in DNA and uracil is present only in RNA. Nucleoside. Each nucleoside consists of sugar molecule and nitrogenous base. I told you sugar plus base is your nucleoside. What is a nucleotide? When phosphate group is added to it, you get your nucleotide. So, nucleotide is phosphate plus sugar plus nitrogenous base. Nitrogen base plus sugar nucleoside, both of them with phosphoric acid is nothing but your nucleotide. This is what your DNA looks like from 5' to 3' end. It's like this, 5' to 3' end. They're anti-parallel to each other. Okay, adenine and forms double bond with thymine and uracil, and cysteine and guanine form triple bond. It has a double helix structure, as you saw already. It is a polymer of deoxynucleotides. The strands are 3' to 5' phosphodiester bond. In both the chains, adenine and thymine are there, 2 hydrogen bonds are there, whereas for guanine and cysteine, 3 hydrogen bonds are there. A always attaches with T in the case of DNA. In the case of RNA, for RNA, A attaches with U. Okay? And C always attaches with G, irrespective of whether it is RNA or DNA. Now, what are the functions of DNA? Self-replication, protein synthesis. These are the two major functions of DNA. Alright, beta. Now, ribonucleic acid is a polymer of purine and pyrimidine. Ribonucleotides are there, linked with 3', 5' phosphodiester bridges of RNA. It basically is single-stranded, beta. It is single-stranded, unlike DNA, which is double-stranded. Alright then, so with this we have come to the end of our session where we have discussed biomolecules in detail. This is a very short and sweet chapter. I hope you were able to understand. However, if you have any doubts, please feel free to leave your doubts in the comment section. Alright, so until we meet next, keep smiling, keep revising and stay healthy, beta. Bye-bye.